So welcome to today's SCItalk webinar. My name is Tim Reynolds. I'm chair of the SCI Public Evening Lecture SCItalks committee. So we arrange or coordinate this initiative. And it's my pleasure to introduce Mark Post, who will be giving today's, which is effectively the second of a triad of food-related talks that are running under SCItalks. As ever, those who are familiar with this format, there will be, do not forget, there will be a question and answer session towards the end. But please, as the talk continues, the best way to make your contribution is to type into the Q&A box within the Zoom function. I believe, in actual fact, for this session, the raise hand function is being disabled, so that will be the only way to get a question into Mark during the talk. And of course, also all attendees are in listen-only mode. So that's the way we're going. So don't forget to put your questions in during the session, and we'll have a session afterwards to put them to Mark. So it is now a pleasure to introduce Mark Post, who is the Chief Scientific Officer of Mosa Meat. He originally became, I think, was trained as a medical person, as a doctor, and came into probably co-founded Mosa Meat maybe about 10 years ago now, or that sort of order. But anyway, I shall hand over to Mark right now, because he knows more about it than I do, and we look forward to your questions and an excellent session. Thank you, Mark.
Thanks, Tim, for the introduction. And it was only 2016 that we started the company, so it's relatively recent. I assume you can see the pointer option.
Yeah.
You can see the screen?
Yeah.
All right. So yeah, thanks for the introduction. Let me explain the title: Cultivated Meat from the Field through Cellular Agriculture to the Plate. And so we call it cultivated meat. It's meat as we know it, but then made in a different way by just doing cell culture and tissue culture from the field, because we still need to feed those cells. So there will still be, and I will allude to that in the talks, there will be food ingredients coming from regular crops but then transformed into a feed for cells instead of for animals or for people. Cellular agriculture is a term that was coined a number of years ago by New Harvest, which basically describes all the activities where foods are being made that traditionally are agricultural, but are now being made by fermentation or cell culture or tissue culture and to the plate, because we intend to make consumer goods. So as a disclaimer, I founded Mosa Meat in 2016. So when this all becomes a success, I also derive some financial benefits from it. And at the same time, I'm still professor of sustainable industrial tissue engineering at Maastricht University. So why are we doing this? And the major reason is this is the meat consumption, the global meat consumption until 2020. And you see that there's a steady rise in meat consumption. Most of the rise comes from Asia, but there's still some increase in South America and of course Africa, and that will only become bigger. And according to the FAO, in 2050, so only 30 years from now, or less even, meat consumption will be 70% higher than it currently is. So globally, meat consumption is on the rise. And if we live in the UK or in the Netherlands, we might get the impression that meat consumption is coming down. And maybe it is slightly, not much, by the way, but slightly, but that's completely offset by the global increase in meat consumption that is to be expected from going from 7 to 10 billion people, but also increasing welfare in China and India and in Africa. And people, when they become richer, they start to eat meat. It's a status symbol in that regard. There's a big relationship between gross domestic product of a country and meat consumption. Um, that is indeed a problem because meat is a very resource intense part of our diet. So for all the meat that we consume and produce, it accounts for 15% of all greenhouse gas emission. It's pretty much the same as the transport sector, it's slightly less. 80% of all the agricultural land that we use actually goes into feeding animals and not directly into people. 20% of our water use, and of course there's a lot of antibiotics that's coming down, but it's still a high volume, and of course animals are being slaughtered. So there are a number of reasons, and currently I don't know how it exactly is in the UK, but in the Netherlands you cannot open a newspaper without a reference to livestock agriculture as a cause of greenhouse gas emission and nitrogen accumulation. So when we had a government-funded project on this because the technologies to do this, to make meat out of stem cells or out of tissue culture, has been around for quite some time, almost 30, 35 years. But nobody really had the stamina to make this happen and to make meat out of this, because everybody thought it was too expensive, it was too inefficient, and for sure, cell culture is not cheap and is not very efficient. So you have to think about what the possibilities are rather than what the limits are. This is somewhat of a strange picture. This is me with a Google Glass on, and the back head here is Sergey Brin, the co-founder of Google. And why is he on this picture? Because he funded that first research that led to the hamburger in 2013. It was very lucky that he, at some point, thought about that this should happen, approached me and said, "Okay, I want to help you." And the first hamburger that we made in 2013 probably cost about, not the hamburger itself, but the whole event and everything around it, probably about 2.5, 3 million. And for somebody like Sergey Brin, that's not a problem. That's not something that you face as a researcher every day. So we were quite lucky to get into contact with Sergey, and he financed the whole endeavor. And again, the technologies were kind of there, but what we wanted to do was to show to the world, this is possible. And by the way, we need to think about how we are going in the future to provide food, and in particular to provide meat. So this is that famous or infamous presentation in 2013 in London. It was essentially born out of frustration because people said, okay, nobody is interested in this, so you don't get funding. Okay, that cannot be. Everybody should be interested in this, so let's stage a public show where we show that this actually is happening right now. You can do this, and also to explain why we were doing this. That was a huge success, much more than we anticipated. And as a result of this presentation, we now have 150 companies worldwide and a number of academic institutions working on this topic. So what is it? It's the idea is actually quite simple. So you take from a cow or from a pig or from any other animal, you take cells, typically out of a muscle. That muscle has stem cells in it. They repair your muscle when it's injured, and that's also true for the cow or the pig or chicken or fish. You can take those cells and you can let them proliferate many, 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 many times. And then you can, because they originally are designed to make tissue, you can let them make tissue in the lab. So you can do that for muscle tissue and for fat tissue, and I will show that we have indeed done both. In the meantime, there are other sources of cells that you can use for this purpose, and some companies are doing that. You can use embryonic stem cells. They have the advantage that they proliferate for even longer than the adult stem cells, but They have the disadvantage that they don't easily differentiate into muscle tissue or fat tissue. Or since the discovery of Yamanaka, you can use induced pluripotent stem cells, which are essentially kind of an embryonic stem cell-like cell, but then derived from adult cells. So the next phase is that you let them proliferate. We know how to do that. Those technologies are around as well. And finally, you let them make tissue, either muscle tissue or fat tissue. Now, this all sounds very simple, and in a way it is. Here it is again. You take a biopsy from a muscle, you separate them into, in our case, muscle cells and fat cells. You let them proliferate. You let the muscle cells become a muscle fiber, and the fat cells become a fat kind of blob, if you like. And then you combine them into a hamburger. But of course, this looks very simple, and in theory it is very simple. In practice, a lot of optimizations need to happen. I already mentioned cell culture is expensive. It's not necessarily very efficient. And the whole purpose of this is to make it cheap and to make it very efficient so that we don't need as much resources as we need for regular beef, and cheap because people may want to pay a little bit more for something that is new and good for the environment and good for their conscience, but that's also limited. So eventually you need to make it cheap. When we started, we thought to make muscle fibers and fat blobs. In the meantime, a number of other approaches have been taken to get to a sort of similar meat component. And if you go down, the biologists among you will understand this picture from a muscle to a muscle bundle, to a muscle fiber, to a muscle cell, and eventually within the muscle cell, where all the contractile proteins are that make the muscle contract. It's a very monotonous piece of tissue, containing high amounts of actin and myosin as the contractile proteins. So what you can do through fermentation, you can just make these proteins, or a protein called myoglobin, which turns our beef red and which is a nutritional source of heme iron. You can do a cell-based approach where you basically only grow the cells and not let them differentiate, and you have animal cell proteins in, for instance, an otherwise plant-based product. You can make fibers like we are doing, let the cells differentiate into muscle tissue or into fat tissue. And finally, you can also make the entire tissue, make let's say, a ribeye steak. And currently we're seeing that some companies are focusing on some of those strategies. So there are diverging approaches. Now, starting with the cells, if you take the cells from a skeletal muscle from a cow, let's say, then of course there are all sorts of cells in there, and you want to specifically focus on the cells that you need. Typically what you do then, if you are in a scientific lab, you know which markers are associated with which cells, and there is a technology called FACS, where you can fluorescently tag these markers and then have a sorting machine that on the basis of fluorescence sorts the cells that have these markers and discards the cells that don't have these markers. And this way you can enrich the cell type that you want. And here on the right, you see that if you do that, your percentage of PEX7 positive cells increases tremendously by approximately 15-fold. PEX7, by the way, is a marker of those muscle stem cells, specifically muscle stem cells, not fat stem cells. This is a very busy slide. I apologize for that. And for those who have never seen a single cell sequencing, this is a relatively new technology where if you have a bunch of cells with different origin, by knowing the expression or by measuring the expression of RNA or proteins in these cells, typically RNA, for every cell, you can get a family of cells, and here on the left on the upper left, you see that every dot here is a single cell with a particular expression of proteins and RNA, and from all these parameters, you can get kind of a fingerprint of every cell type. So here we have our fat cell population in blue, and we have our muscle cell population in yellow. And you already see that these populations are also not completely homogeneous. There are still some other cells in there with different expression patterns. So we are not fully specifying the cells yet, not fully specific for the cells, but we are getting better and better. And that's important because you don't want contamination of your cells with other cell types, because you really want to make muscle in one part and fat in the other. One important feature that has been mentioned many times, especially in the popular press, but also in the scientific press, is traditionally for cell culture, we use serum. Serum is a blood-derived product. It's coming from cows, by the way. And about 140 years ago, when people started for the first time being successful in culturing cells outside of the body, they became successful because they added serum to it. That is a product that comes from the cows. It's kind of an ugly way of harvesting it. It's not very stable. There are all sorts of issues with it. So one of the first challenges was to get rid of serum and to replace that with a set of proteins that we call growth factors. There are many growth factors in our body. The most known one is FGF2, which is a stimulant for proliferation for pretty much every cell that we have in the body. VEGF is another one specific for blood vessels, and so there are many of those. And what you do is find for your particular cell type the exact cocktail that you need to replace serum. And once you do that, you can create what we call serum-free medium that in most cases is actually more effective than serum-containing medium itself. Medium, by the way, is the soup in which we grow the cells. It has all the nutrients, it has glucose, it has vitamins, minerals, everything that the cell needs to grow. So we have succeeded in developing a number of those media different for muscle cells, different for fat cells, different for proliferation, and different for differentiation. So, but it's entirely doable. It just takes time and effort. And here you see that this is what you have to do as a company. At some point, you have to make sure that this is a robust thing that you can, for every biopsy that you get out of a cow, you can actually get a large number of cells so that their proliferative capacity is pretty much the same and guaranteed. That's not quite true, but you want to get there as much as you can. And so you see here, if you just focus on the left upper graph here, you see here the cumulative population doubling. So a population doubling is whenever the cell doubles, right? Because it divides, it doubles in number. And here are the days of culture. And you see that not all of them, some of them actually stop after 40 days and after about 25 doublings. And a lot of them, or half of them, kind of continue up till 40 or even 50 doublings. And that was the 50 doublings, what at some point hypothesized by a German called Leonard Havlik. He said most cells actually can double about 50 times. And we show that that's true for about half of our biopsies, so half of our cows, if you like. Now, if you do that, then you can calculate how much beef you can actually get out of a biopsy. The biopsy is typically about half a gram, so it's very small. And if you get to 32 doublings, you get about 2,000 kilos of beef out of them. So that means that you would, and typically out of a cow, you get only 250 kilos out of one cow. You can take more than 20 biopsies out of a cow, so you can see that you can gradually reduce the number of cows that you need to create this type of meat. And that's exactly what the purpose is. You can even be a little bit better than that, and trying to see if those cows that don't do very well, if you can extend the number of doublings that you get out of the cells, and you can do that. This is a compound called a P38 inhibitor, was also a known technology. And if you do that, you can see here that on the PAC7 positivity, which is the stem cell marker and an indication of how much they can proliferate, that keeps on at a higher level when you use that P38 inhibitor here in the black boxes. And then you look at the cell numbers, the proliferation actually short term is the same, so it doesn't affect the cell numbers. Short term, long term, they continue for a longer proliferation and also for a longer differentiation. So this is kind of the age of the cell. And if you don't use that P38 gradually after a number of population doublings, they go down. If you use that P38 inhibitor, you can keep that differentiation pattern kind of at the same level. It still goes down a little bit, but not as much. So we need to do all these optimizations to make sufficient product out of a single biopsy. Now, that's for proliferation. And for differentiation, making them into muscle tissue, that kind of goes spontaneous. Once you take out all those stimuli to let them proliferate, they start to differentiate. And the differentiation of a muscle cell is a very interesting process. First, they need to merge. So a muscle cell is a merger of typically 100 cells. So they merge, they make one big fiber, and then they start to contract. And they start to build up all their contractile proteins. And if you do that with a number in parallel, you get a muscle fiber. Now, in order to enhance that, you can see, okay, what happens exactly during that differentiation? And are there things that we can enhance? So this is, again, an RNA sequencing where we have looked at expression of certain proteins, and every dot here in this case is a protein or a piece of RNA rather. And everything that's on the right is increased during differentiation, and everything that's on the left is decreased during differentiation. And if you then look at some of those targets, like this is an IGF-1 receptor, it's one of those growth factors, the receptor of it. This is the oxytocin receptor. So there are a number of receptors that you can find here, and each of these receptors you can target with specific peptides and see if we stimulate them, do we stimulate differentiation even more? And indeed, that's what you see. This is with serum, and this is with serum-free. I need to explain what this is. Every green thread here is a merger of muscle cells. So the more of those green threads, and every blue dot in the middle, that's not easy to see, but every blue dot in the middle is a cell that does not participate in this merger. So eventually you want to get as many of these green strands as you can to make the process optimized. And with serum-free, with all these optimizations, you can actually do that. What is interesting, I showed those blue dots in the middle, and here you see it even better in black and white. So here are the muscle fibers, and in between you see those black dots. Those are cells that refuse to, that's the refuse to fuse. And the question is, can they fuse, or they just don't do it? And the fact is actually that they can, but they don't do it. And we know that because once we take out those muscle fibers, those primitive muscle fibers, the cells that are left behind go through a other differentiation route. So they start to fuse. Once those fuse cells are out of the way, they start to fuse. So they can fuse, they just don't do all of that. So that we also wanted to optimize, because if only 40% of the cells fuse or 50%, obviously it's kind of a waste. So we have gone again through a large number of interventions with agents that we add to the cells, and then we can actually coerce 95% of those cells to fuse and to make muscle fibers. All right. Long story short, currently this is now not in green, but in red, and you see that pretty much all the cells participate in fusion. That leads to a protein composition of the muscle tissue that is very similar to that of minced meat that we just got from the grocery store. So protein level wise, we are kind of at the same level of where we need to be. This is also a very technical slide, but it's an important issue because protein content alone is not the only thing that you want to achieve. You also want to have the specific muscle proteins. And myoglobin is a particular good example of that. That again is the protein that stains the beef red, and it's also a nutritional source of heme iron. And here you can see that in this part, this is what we see in normal beef, and this is what we see in the beef that we have made. So it's not quite the same yet as normal beef, but it is up there. And this is about the same, myoglobin level as pork. Pork is pink and not red, so it has less of this myoglobin, and this is about the same level of pork. That was for the muscle tissue. For the fat tissue, we also do the selection. This is a very similar slide to the one that I showed for the muscle, and you don't need to look at all these panels, I guess. But the bottom line is that we also can find specific markers for the fat population. It comes from that same biopsy. And so we separate the cells into fat cells and satellite and muscle cells and cells that are neither. And here we have also spent a lot of time, is this a heterogeneous population or a homogeneous population? And we were quite lucky, and we are quite lucky that very early on we could actually get 90% of all these cells to differentiate into fat tissue. So here is this is what fat tissue looks like. It is these fat is all the fat is green stained here, and these are these big bubbles of fat, and that's typical for a white fat cell. It takes actually quite a bit of time to make fat cells, more than to make muscle cells. Muscle cells 10 days, fat cells about 24 days or 28 days, as you see here, and then you gradually see increase the amount of lipid fat in that cell. It's also true fat cells. It's not cells that are filled with fat, but it's true fat cells. They make the fat actually themselves, mostly out of glucose and out of amino acids. And we were even more fortunate. This is, by the way, this is the fat that we have cultured here in the middle, and this is fat that we get right out of a cow. And we were very fortunate to see that the fatty acid composition of that fat is almost exactly the same as that of conventional fat. This is depicted in this slide. This is day 0 to day 28. So over time, the fatty acid composition reflected here by the color coding changes, and then it stabilizes at day 28, actually already a little bit earlier. And then here you see the fat composition of fat that we get out of the cow. And the color coding actually refers to the fatty acids that are in there. Every fatty acid has a particular color. So the composition is quite similar, and is much more similar, of course, to conventional fat than, for instance, coconut oil, which is typically used in plant-based products. What we do see that's also somewhat lucky is that the amount of unsaturated fatty acids that are better for you are lowering your cholesterol is actually slightly higher in the fat that we have made than in conventional fat. Again, this is kind of a lucky stroke. It's nothing you can design it, but we actually didn't. This was the first outcome. So that's all the kind of basic science, but then of course you need to start making a product, and for that you have to scale up production. That's a completely different area of development, if you like. So you want from a cell culture dish go to a big tank, actually a 10,000 or maybe even 100,000 liter tank. Some companies are dreaming about that right now. But the cells need to grow on a surface. They cannot grow just freely in the liquid. So you need to grow them on a surface. That's why most people use microcarriers. These are little spheres that are sitting in the soup, in the medium, and the cells grow onto these spheres. That's very helpful. It works very well. We have tested a large number of these spheres that are out there. They all work quite nicely. So this is one of those spheres, and all the little dots are the cells that are sitting on there on it. And if you wait a couple of days, eventually the cells become completely colonized by the spheres become completely colonized by cells. Now that is nice, and it works actually very well, but the spheres are somewhat cumbersome because they take up space in the tank, and that is space that you cannot use for something else, for cells, for instance. And also you have to separate the cells from the spheres. So what we are currently doing is we grow the cells in aggregates. Remember, they have to stick to something, but they can also stick to each other, and that way they grow. So this makes it easier. You don't have to separate the spheres. We call this aggregates, and you can get much higher cell densities with the aggregates. So the process looks then like this, that you start with a very small number of cells that are taken from a cryobank. Then you use increasingly larger systems, and you end up with these 1,000-liter tanks. That's where we currently are at. Some people, again, are fantasizing about using 10,000 liters or even higher. But currently we are at 1,000 liters. And if you do sort of a calculation, a rough calculation, how many tanks you would need to replace the entire meat production in the world, then you would need 20 million of those tanks. That's a lot, but it's not undoable. It's roughly, to give you a perspective, 1.5 times the current fermentation capacity in the world. So it's not outrageous. It's a lot, but it's not outrageous. So the scaling up is something that, at least for the cells, can be done. I don't talk about the scaling up of the tissue, but we are doing it roughly in the same way. Then, of course, the next thing is that it has to be cheap. And currently, all the components of the cell culture things that we use are expensive because they are pharmaceutical grade. They come from the pharma industry or from the medical industry. And we need to get rid of that because we are making a food. We are not making a medical product. And the most expensive component of the whole process is actually the feed. This is true for the cells, but it's also true for cows. For cows also, the most expensive part is actually the feed for the cows. And if you kind of decompose what's in there in that feed, I mentioned already, it's glucose, amino acids, minerals, vitamins, some of those growth factors that we use to replace serum. And eventually you want to economize how much of that you use. So here you have a list of amino acids. On the left, you have the essential amino acids that the cells actually do need from the feed. On the right, you have the non-essential amino acids. Those are all the amino acids that you probably should give, but if not, the cells can actually make them themselves. And if you culture the cells for 7 days, which is already a long time, you see that some of those amino acids are still there. They are not depleted. And if you throw away the medium, then obviously it's a cost factor. So what we are currently doing is we keep the cells as long as we can in that medium and just replenish the amino acids that are being depleted. Same for sugar, same for glucose, same for other components. So we basically supplement everything that they need, and we don't replace the medium entirely. And that way you make it and more resource efficient and more cost effective. So to just give you an idea, in the last 2 years, where what our growth, the cost reduction was, starting at 100% and now at 1.5. So we achieved a 98.5% reduction in cost of the medium by changing by feeding those depleted amino acids, by replacing some of the factors by cheaper factors. And we still can go much further than this. And this is where other ingredients come in, if you like, and we are playing increasingly with those ingredients. So this is kind of a indication where the price per kilo of this product over time will go through, depending on a number of scenarios and strategies that we are following. And then eventually we want to achieve like a €6 or a pound per kilo of this product. And we know that we can get there. It will just take time. This is just a fun slide in between. If you would, so currently we're still using some pharma grade components to produce this. And if you would make chocolate chip cookies with pharma grade components, they also would be very expensive. So 25 cookies, $30,000. So it doesn't make any sense to use pharma grade components for this. So here what we have done is we used hydrolysates, pea and wheat and all sorts of hydrolysates, and feed them directly to the cells, not worrying about what exactly the composition is, and just see whether the cells thrive. And here everything that reaches 100 is basically the same as our pharmaceutical grade medium. So some of those hydrolysates are actually working quite well as a medium supplement. So we're currently working with a big feed component supplier to see which hydrolysates and which amino acids from feed grade we can actually use in this material. Now, finally, what I realized, if you or learned, to be honest, when you do something like this, you have to deal with public perception. And in the beginning, when we introduced this in London, the popular press said, yuck. There was some reservation, some immediate response, oh, maybe we shouldn't want this. And it was kind of puzzling because, nobody had tasted it, or at least very few people. And so it was kind of puzzling where this came from. And I think this is generally, if something completely new, a new technology, whether it's GMO or some hybrids or cultivated meat, there is a initial reservation, and there's a lot of literature on this, by the way, so it's kind of understandable. In the meantime, a lot of surveys have been done, asking questions. Do you accept this? Do you want to eat this? And over time, we see an increase in acceptance, so that's a good thing. What we did, I don't have a lot of time to go through this slide, but what we did our own study where we basically asked the question, what kind of information drives people to accept this? Because a lot of people said, well, people are only interested in, whether there are personal benefits, health, or whatever. So we divided the group in three. Some got information about the societal benefits, some on the personal benefits, and some on, it's just good meat. And then, because it was organized together with a student from Dijon in France, we also decided to have people taste 2 pieces of hamburger, one that was labeled conventional and then that one that was labeled cultivated. And since it was not really allowed to feed people cultivated meat yet, and it still is not, we had to make the 2 pieces essentially the same from the same hamburger. So the people thought you can apparently do this in a social psychology study. The people thought they were eating a cultivated piece, but they actually ate a regular piece, just as the same as the other one. And we let them comment on the taste and the texture and then the flavor and things like that. So what we found was kind of interesting. First of all, it was not really distinctive, the result of which information we gave. So we asked the people, are you in favor of this? Do you want to taste it? Do you want to buy it? Do you want to replace your current meat consumption by cultivated meat consumption? And you see that, you know, the color coding refers to that piece of information that we gave, and it's pretty much the same. So whatever information you give doesn't really matter that much. What does matter, because we asked the questions before and after we give the information, is that you give information. So here you see from before we gave the info to after we gave the info, that you see an increase in for all these questions in acceptance. And we see that throughout the surveys, that as long as people understand and know what this is, they react to these questions in a much more positive manner than if they don't know yet. And if you want to know what the reaction to the taste was, it was somewhat predictable that people found the cultivated meat piece taste better than the conventional one. And we kind of know that if you put very expensive or very cheap wine in an expensive bottle, it tastes a lot better. So there's a lot of influencing of taste going on here. So, as I mentioned, the 2013 presentation did a big thing. It created this industry, and there are now 150 companies worldwide with a gravitation in on the West Coast, in Western Europe, Israel, and somewhat in Asia. Singapore is now a kind of a bigger focus. So worldwide, this is becoming a somewhat bigger industry. They're all startups, so it's still not extremely big. But in this year, we will see the start of a number of factories. A factory in 2 factories in Singapore, a factory in Israel and in Europe, 2 factories in the US. So we will see gradually some of these factories coming up. Capacity to produce still very small, but it is happening. And it also requires regulatory approval. But first, if we do this, does it actually matter? Is it more efficient? Is it associated with less greenhouse gas emission and so forth? And this is also here, there are a number of studies. They are quite anticipatory because there's no real factory yet. But you see here, this is the most recent and most elaborate study from CE Delft. And this is a graph where it's where the compound environmental impact is shown, and it's compound by global warming, land use, water consumption, fine particle formation, human toxicity, and other, like antibiotics, for instance. And you see that invariably in all these studies, beef and dairy are the worst. They have the biggest impact on the environment. There's no question. Pork and chicken are a lot better. Plant-based diet is the best. There's no question. And here you see 2 interesting ones. This is cultivated meat, conventional and sustainable. And so if you would do this the conventional way, it would be in between pork and beef. Okay, not perfect, but okay. This is, by the way, if you use traditional fossil fuels to energize the system. If you use renewable energy, you arrive between chicken and plant-based. And of course, you know, once this comes online in the next 10, 15 years, chance that this is renewable energy is extremely high. I wouldn't want it a different way. So this is a novel food, meaning that it has to be regulated in all geographies. Also in the UK, the Food Safety Authority has to look at the data, has to be sure that this is safe, and then it can enter the market. And this has to be done in every country or every geography, which is kind of painful. Europe is one, but the USDA is the US is different. Singapore is different. UK now is different. So you have to go through all of these, which is a little bit weird because if something is safe for somebody in London, it's also safe for somebody in Singapore, I would imagine. But anyway, this is how it is currently. There are now 3 products, one product really approved in Singapore. It's a chicken nugget. And there are 2 products that received a no questions letter from the FDA, so they can further go along with the USDA to introduce this into the market. How much time that will take, we don't know, but the first step is taken. I am based in Europe, so we have to deal with the European Food Safety Authority, yeah, they take a little bit longer because after the scientific part of the dossier evaluation, there's also a political part, which, as we know in Europe, takes just longer than pretty much anywhere else. But to their credit, they did actually specify what the process was way before anybody else did. And pretty much everybody is using the guidelines and the detailed requests that the European Union has identified already 8 years ago. So with that, I would like to conclude. I hope that I've given you an insight in what this is, why we are doing this, where we are at the moment, and for sure, it's still some ways to go to get it to price parity, so to get it to market price, to get it into the supermarkets, and to get it to the same quality that we're used to with regular meat. But it has all that potential, and I don't think we can afford to wait until meat consumption becomes so detrimental to our environment that we have to really make hard choices. So with that, I would like to end and happy to entertain questions.
Well, thank you very much, Mark, for that. That was fascinating, in actual fact. We have some questions for you. I'm going to take chairman's priority and ask something. The presentation was focused very much on beef, I guess. Well, beef substitute, but I guess that's to do with the value proposition in that beef is relatively expensive compared to pork and chicken. But then I see that the chicken product is in Singapore. I mean, is there any difference in sort of how much it costs to make a chickeny pork type?
Not really. Not really. And I don't want to criticize my competitors, but chicken so we are doing this primarily from an environmental point of view, from an environmental perspective, and chicken, as you've seen in that graph, is actually not that bad. It's of all the animal proteins, it's probably one of the best. Actually, fish is even better, but that has different issues. But yeah. So from that perspective, and also from a cost perspective, it is not the most obvious to do chicken. From an animal welfare perspective, it is.
Okay. Yeah. Right? Yeah. Okay. Well, a similar sort of question from one of our attendees is, how much control do you have over nutritional value of the cultivated meat, and are there possibilities for creating healthier meat?
Yeah, it's a good question, and I kind of alluded to it with the fat component, that we happen to have more unsaturated fatty acids in our fat, which should lead to a cholesterol reduction in the people who eat it, and therefore a lower chance of cardiovascular disease. I
Okay.
say should because that all would need to be established. Meat is not an unhealthy food. The fat is to some degree unhealthy because it's saturated, but it's not an unhealthy food. There are two health problems associated. One is, again, the fat, and the other is a slightly increased risk for colorectal cancer.
Yeah.
And that's why the World Health Organization has now issued that you should eat less than 300 grams of red meat per week, which is not a lot.
No,
That's,
it's yeah, a
which
couple of
is really
burgers.
not a lot. And the culprit is not very well defined because it's red meat. It's tempting to associate that with myoglobin, for instance.
Yeah.
And I have not emphasized that, but in that myoglobin graph that I showed, it's actually implied that we can titrate the amount of myoglobin that is in that meat.
Ah,
So we can make it more pink, we can make it more red, we can make it even paler, so that if that's indeed the culprit of the increased risk for colorectal cancer, we can actually do something about it. But other than that, our first focus is to just replicate its traditional nutritional value because that's good enough.
yeah. Excellent. Yeah. I mean, I guess if you sort of titrated out the myoglobin, you'd end up with something more veal-like in actual fact.
Mm,
Which, yeah. So,
yeah, exactly. Yeah.
okay, that's interesting. Yeah. Another question: do you, well, I think you come to a certain extent this. Do you expect any significant challenges relating to regulatory science? What regulations have to be fulfilled for cultivated meat to be allowed to be sold in stores? I mean, that's EFSA, yeah.
Yeah, that's EFSA, and again, those guidelines are pretty well defined, and so every company has to file a dossier with compositional analysis, with tracings of where all the ingredients come from, with microbiology analysis, all the analysis that you can think of that eventually could affect safety of consumers. And there are some new ones that are specifically associated with this technology, like the use of growth factors, for instance, to replace serum. And there will be specific focus on these novel kind of ingredients, if you like. We recently have, the FAO recently has organized a big meeting, a 4-day meeting with experts on try to kind of harmonize the sort of risk analysis that are associated with each of these, with the production system and with the ingredients that are being used. And so they will come out soon with a very detailed report. I was part of that committee, with a detailed report of where the experts think that the focus should be in terms of regulatory control. And where some things that have been expressed as a risk are so unlikely that they are on par with, just choking on a piece of conventional beef.
Yeah, which you can also do that with tofu, of course. So,
Right, exactly.
A question which you may not have the answer to: are the Singapore's chicken nuggets affordable?
No, depends on what you call affordable. It is for 25 grams. I think it's 50 Singapore dollars.
Okay. Yeah.
So affordable if you want to eat only 2 pieces. If you want an entire meal, then you better be an inhabitant of Singapore.
Yeah, indeed. Yeah. Other one.
So this is not really a market yet. This is just a introduction to tell the story and to feel the waters, and that's basically what it is.
Well, yeah. I mean, you can spend an awful lot of money on a steak in some places.
Yeah.
I mean, yeah. And I think you're paying for, you're showing you can pay the money. When can we say that lab-grown meat has improved animal welfare. How far are we from that? We're there already, aren't we? In that we are taking a biopsy, which is not a
Yeah. So we've actually published this. We call this the multiplication factor. So how many cow equivalents can you get out of one cow through this? And this is 165. So that means that with the current state, we would reduce the total number of cows by a factor 165. So roughly, which may, if you think about it, going from 1 billion cows to only a couple of million, may for some people be not enough. From a food security and environmental perspective, it's a big deal.
Yeah. Okay. Another question of it. Interesting. I think I've seen things on this. Have you thought about how this might be combined with 3D printing, which is also being used to make new food?
Yes. Yeah. Of course, from tissue engineering, if you come from tissue engineering, you always think about 3D printing, and so we see a lot of new developments in 3D printing that make it scalable and fast. That was originally the Achilles heel. What I've shown is that how we make small tissues, minced meat portions, actually. If you want to make a ribeye steak, you would have to do something like 3D printing to at least form the lattice, the scaffold to which the cells kind of adapt. And so yes, there is a lot of activity there at different levels. One level is preparing for the ribeye steak. One is using these chunks of meat and a enzyme called transglutaminase to actually piece them together and create something that looks like a steak. That also has been done. So it's being considered at different levels.
Okay, so I mean, in terms of timelines for these sort of things, your cultivated mince may arrive in the store, and a ribeye may arrive fairly soon afterwards, or in a similar sort of time frame, or are we talking
No, no, it's going to be later, I think. Realistically, 5-6 years later, because it's technically a bit more challenging.
Yeah. I think it's a bit of a technical question. On the sensory acceptability presented earlier, what was the sensory technique used? But it wasn't actually,
No, it was just tasting by the applicants. And they commented on first visual, smell, texture, taste, juiciness, the traditional factors.
Yeah. And I guess you probably answer this very quickly. You can use a milk cow as a source of the stem cells, of course.
Yeah, you can. Yeah. And we have done that. We have done many strains of cows to see if there are sort of preferential strains or preferential sites of the cow to take it from. It's pretty similar. The differences in look of the cow and amount of meat that they have is vastly different. But if you reduce it to cell culture and tissue culture, the difference disappear largely.
Okay, yeah. Maybe come to the final two here, because I think we're running out of time. I think we've got to close at on the hour, which we're very close to. Oh, never mind. If the goal is mainly environmental or emissions focused, do you personally think that encouraging people to eat cultivated meat is going to be easier than encourage them to eat essentially a plant-based diet?
Yeah, I think so. That's the reason why we're doing this. If I had believed that it would be easy to change behavior of consumers away from meat consumption, I would not do this. Then, because that's always, as I indicated, kind of a better approach in terms of outcome. But I think it will be just hard. And in fact, I think that these somewhat intermediate products from a cultural perspective will actually help the transition to a more plant-based diet. I hope it
Yes.
does.
Yes, part of a part of a journey, so to speak. Yeah.
Right.
Yeah.
Right.
One final question, basically. Lamia says, thank you very much. Very interesting. Do you have any literature to recommend on this subject area? Is there anything? I mean,
Right. Yeah, there's a book from Paul Shapiro called
okay.
yeah.
Yeah.
I'm bad at titles. I know the author very well, but that's a very nice book actually. He's a good storyteller, and that's probably the most interesting at the moment. It's a little bit older, and there are now two books coming out, but they are really scientific, so they are really going into the science. One is actually edited by me, and the other is edited by a professor in Canada.
Okay, excellent. Well, Mark, thank you very much for your talk. It's absolutely fascinating. And I think I said earlier, with the IPCC report that came out yesterday, I think we all need to really, really think about what we're doing with our lives and our carbon footprints, etc. Otherwise, things are not going to go well at all. So I think I need to, well, thank you once again. And
Welcome.
a quick reminder about, if you're not a member of SCI, please do think about joining for its various benefits. Future events for SCI, as Factor of the Future at the end of March, SCI Day of Science and Careers in April, and our next SCItalk, which is the third of our triad of food related talks, is on the 26th of April. So do sign up for that. It's with Dave Hughes of Syngenta on food foresight and future of farming, which I think we've just heard a lot about actually. It's going to be in a fermentation rather than a farming, but maybe not all. But anyway, thank you all very much for attending, and hopefully we see you again in April. And once again, thanks to Mark.